All right, let's get started. So I want to make sure to welcome everybody to our CRISPR office hours. And today's talk is about how biotech companies can keep calm and carry on during the COVID-19 crisis. Before we get started, I want to take care of a few housekeeping things. All the CRISPR office hours are recorded, and we have past ones available on YouTube, and this will be available afterwards on YouTube as well. As we've mentioned a few times for returning folks and for new folks, CRISPR Office Hours was developed as a way for the community to interact with one another during this crisis and understand how we can all tackle the issues we're seeing and get through it together and ask questions and make it as interactive as possible. Our goal at Synthego is to merely host these and help build a community and let the community hear from other genome engineers, researchers, and scientists, as well as life science executives, to understand the impact of COVID-19 on research, as well as seeing how we can get through this. So during this session, the more interaction, the better, the more value we'll all get from this. So send in your questions using the chat and we'll be sure to get to them. And if we don't get to any of them, please don't be offended. We'll be sending, you can email me av@synthego.com if you have any questions that weren't answered, and we'll try to get to them either email or in the next office hour session. So real quick, my name is Aditya Vempati, and I'm the VP of Marketing at Synthego. And I'm really excited about our panel today. To kick things off, I want to introduce my co-host, Kevin Holden.
Hey everybody, coming to you live again from the CRISPR Office Hours studio. All right, so as everyone saw from our title today, the topic of today's discussion is to keep calm and carry on. As I'm sure most of you know, this historical slogan has become very popular in the last few decades. And we feel that it's actually never been more appropriate than it is now. And we'd just like to remind you all that at CRISPR Office Hours, to keep calm and CRISPR on. This is our version of the famous poster. And it turns out that the whole idea of keep calm and carry on wasn't the only slogan in this public assurance campaign. So actually there were 3 posters that were made by the Ministry of Information and by the British government at the start of World War II. And these were posted all across England, from underground stations to parks to factories. And really their purpose was to strengthen public morale in the face of a terrible danger, like civilian bombardments. The author of one of the signs, the one you see on the right, that starts off with "Your Courage," stated that he wanted to develop kind of like a rallying cry that will bring out the best in every one of us, and also put us in kind of like an offensive mood at the same time. And we just thought these were absolutely great. So we've made a few updates to the posters that we think are very appropriate for the terrible danger we're all facing now. Experiments are in peril. Make sure that you work from home with all your might. And really the last one we felt, the courage, was that it's your courage, it's your research, and it's going to be your breakthroughs that will bring us victory against the COVID-19 pandemic.
So thanks for that history lesson. It's always awesome. We get little bits and nuggets every time at CRISPR Office Hours. This time with a history lesson about what exactly was done in World War II and the British era. So what I want to do is let everyone know, everyone who's attended CRISPR Office Hours today is going to be getting a Keep Calm and CRISPR On shirt. Stay tuned to the very end to know how you can get your shirt. And so let's get through. As I introduced my co-host, Kevin Holden, I'm really excited to introduce Ted Tisch, CEO of Mammoth Biosciences. You want to go ahead and say a few words, Ted?
Yeah, thanks, Aditya. Great to be here, and really appreciate the time and the effort. This CRISPR Office Hours is amazing. Watched the past couple of them and really help inform people. If I am the CEO at Mammoth and can talk through a little bit of that later on, but also have deep roots in Synthego and was there for 5 years and really impressed with what has been going on. So I'm definitely feeling calm right now, and I'm ready to get my CRISPR on. So let's move ahead.
So we've done introductions. As we start every CRISPR Office Hours, we like to have a poll to understand how people are doing. It's very much informal, lax, but we want to get, people in the mood of interacting as well as just having conversations. So today's poll is, what is your preferred work from home location? Do you work from the couch, or do you curl up in bed, or do you munch on snacks and sit on the kitchen table, or do you enjoy nature and sit in your backyard? Or do you have a home office setup? And of course, for the folks who are calling in from tropical locations, I'm sure hammock is an option. I know for myself, I actually find myself in the kitchen dining table as I live in a smaller location, high rise in the city. So space is hard to come by. So me and my fiance make do with what we have. For everyone else on the line, send in your thoughts through the group chat. What about yourself, Ted and Kevin? Where do you guys prefer to work from?
Well, I have a house that has a few rooms, not too many, but we have 4 working people in the house. So I'm in our spare bedroom, which is my office now. Pretty happy with that. Probably better than some people. I prefer a hammock. I really do prefer a hammock. That'd be great.
On a beach somewhere. I'm all about working from the couch while I can, so I'm enjoying it. I may be joined by a cat at some point, but yeah, the couch is definitely a little more comfortable than working at a desk.
Oh man, we got a really interactive audience. This is awesome. A lot of folks are saying actually couch and dining table. Those seem to be the ones that are jumping out. A funny one that just came across is from Marble Marquez. I tried my bed, but ended up napping.
That's a dangerous one.
That's a dangerous one. So with that, we want to move on. Thank you for all your responses to talk about the Golden Interim. Go ahead, Kevin.
Yeah, so just to kind of reorientate everybody, for the past few weeks, we've been discussing this model that was developed by the Linus Group, where they surveyed over 2,000 scientists about their thoughts and activities during the pandemic. And so, as you can see from this model they built, we're actually currently still persisting through this Golden Interim phase in the middle here. And so last week we heard from an academic lab at UCSF about how they're coping and also how they've actually been involved with some COVID-19 research as well. So today, we thought we'd bring in a chief of industry, a captain of industry, and discuss how biotech companies can keep calm and carry on during the COVID-19 crisis.
So, Ted, I want to ask you, as CEO of Mammoth, how are you guys adjusting operations at Mammoth during this crisis/pandemic?
Yeah, thanks, Aditya. So this is a real interesting time, and I think you really got to look, when you look at the non-academic area, where you look at companies and the reaction. And I've done a little soft polling of my friends in industry, ranging from like us, a 30-person company in South San Francisco, all the way to large companies that are around the world and global. Everyone's responding a little differently, but the main common theme is safety and social distancing. So it's really come to a point of making decisions around the safety of the employees. And that's been unilateral. And I think this is maybe the biology, healthcare kind of mantra and anchor we have, that we really want people to be safe. So for us, as a small company that's actually in the middle of, I'd say, driving a COVID assay, which I'll share later, there's this real dilemma people are having. And the challenge is how do you be safe? How do you actually operate in a lab where typically you put 3 people in a preamp room. Now you can only get 1 person there because you can only have 1 person within 6 feet. The PPE is really good, typically, if you have it, but you still need to keep that spacing. And so all of a sudden, lab occupancy went from 100% down to 25%. So you took a big hit there. The other thing that I think is more important is that in our company, we made working in the lab, and for those people in the lab, obviously office people and staff people are working from home. But it's optional, no questions asked. If you have an at-risk person at home and you're going home to that person, we really don't want you, you don't want to go and potentially infect them or expose them. And so we want people to stay home in that case, but no questions asked from the management. We just, if you show up if you want, don't show up if you can't. And we'll do this golden interim, which is writing, analysis, and planning. There's a lot of good research going on. I think we got caught up on a lot of our paperwork and reading. And so I think it's important. But in the meantime, the engine's going, and we're trying to make adjustments. We're trying to restack the lab so that we have social distancing, moving biosafety cabinets around. And so there's this careful balance. What I think is interesting this interim is trying to keep moving and trying to keep progress going without putting people in an unsafe environment.
Yeah, that's a good point. Safety always comes first, and making sure people are comfortable in their environment. So what are some of the surprises you guys have come across during this adjustment period?
Well, I think, so probably everyone can agree, communication and social interaction has been limited. And even with the best tools in the world of Slack and text and email and Zoom and video conferences, it's been there's still challenges of having that human interaction. And by nature, scientists are social beings in some ways, and working in a lab with other people and kind of brainstorming in the aisles, as you all know, or sitting across from each other in a desk or next to each other and kind of like, I'm stuck in this problem, help me get out of it. You have none of that really, just at the moment type of innovation, iteration, brainstorming type of interaction. I think I'm not surprised by it so much that it's not there, or it's hard to kind of to create in this environment. It's just it takes more effort to get there. And I'm surprised at the effort people are doing to go there, not in a bad way, in a good way, that people are actually using things like Donut on Slack or other tools to kind of really communicate. We have a video mandatory, 30 people, we're not gonna pull the internet down, but mandatory video on every call, which requires some facial response interaction, so you're not talking into the void.
Cool, that makes a lot of sense. And speaking of interaction, folks, please send any questions you have. We'll try to answer them as we go forward.
Yeah, so this was an interesting piece of data actually from the Linus report, where they showed that scientists in academia were feeling less productive and actually more affected by the pandemic than those in industry. Ted, so companies like Mammoth and Synthego, obviously we're doing some things that are deemed essential to keep scientific progress going throughout this crisis, whether it's from reagents to cells and diagnostics, from manufacturing example. Can you discuss how Mammoth can actually operate efficiently and also maintain that safety for its workers during this time?
Yeah, so we put in some pretty draconian scheduling efforts, I would say. We're basically an open lab where people can come anytime. We're in South San Francisco on the Verily campus, and it's pretty well supported 24/7. So people could come in and work when they wanted to, and have be really productive when they needed. Now we needed to, as I said, scheduling is really critical, and so it took some time to figure that out. I think all of this is new, right? So it creates stress because it's new. We're out of our habits. We're a creature of habits. And so we actually are now scheduling within half. So like, hey, I'm going to be in the office at this point. We can only let 4 people in the lab, lab A have 4 people. And to book up. And what we found out though, they weren't in the lab 4 hours. They were in the lab an hour and 3 hours in the office. And so that made it really challenging to be productive. So then we decided to move it into very agile thinking, move it into half-hour chunks and be a little more prescriptive. And so people have been doing a lot more planning at night, the night before they're doing work, instead of doing it in the morning. So when they come in, they don't occupy space that they're not utilizing. And so that's how I think that's the key thing around productivity in terms of in the lab. And then those people that are not in the lab, constant interaction with their supervisor, manager, or their peers around the work they need. So actually the people in the lab can just do the lab work and respond, and then they're getting feedback from the people that are outside the lab on data analysis for them, so you don't have to spend a lot of time. So it's been interesting. We're still figuring it out. I think we'll be figuring out for 6 months, but we're still figuring it out. But I think that that's a lot of it is just having that, trying to keep that scheduling important. And we've also opened it up again to, scheduling from basically 6 in the morning all the way till 10 at night. Though those times are not as busy as normal.
So I guess obviously implementing this kind of plan really, requires the buy-in of your executive team. I'm guessing also your founders are very supportive as well. Maybe you can just mention that. But and I guess also, it for your employees too. So was this something that you guys decided very early on, to go to them and say, hey, this is what we're going to implement, just, before, and not being so reactive?
Yeah, I wish we all could know we were going to be here right now, a couple months ago. I think we all kind of wish that. I have this thing that became really apparent, and I was talking to my mom, of all people, and she goes, I don't understand this outbreak and this pandemic crisis. Why are we, this was back in, early, mid-February. And I don't understand how it's going up so fast and that. And I said, because Mom, we don't think in log, we think linear. Most people in the world think linear, right? And scientists, actually cell biologists, really think in log because they count a lot of cells and they're thinking about log scale, but scientists tend to be log people. And so I think the adjustment for scientists is actually pretty natural to think around, okay, this is a pandemic, we're gonna have log scale growth of infections, we're gonna have to respond appropriately. But it's, for the general public, I think it's hard to kind of make that leap from linear thinking to exponential. And so it all caught us a little, even though we're scientists, from a management business side, I think a little by surprise. But what I was really impressed by, especially in early February, and again, we're on the Google Verily campus, is that there was a lot of early response by that team, which we could just dovetail on. And I would say that I was really impressed with Verily's ability to really go fast. If you looked at a lot of the tech firms, of course, a lot of those people code at home. They don't have a lab part, but we're really quick to respond to work from home and try to keep social distancing. But when you're in the lab, it's harder. So we just kind of evolved our way through to kind of like, yes, we now need to clean everything and be careful. And then the next couple of days, and we were like, if there was coronavirus detected anywhere on campus or anybody we know, then we're just shutting down and going to where we are now, just being much more careful. So I think I'd like to say we did a good job, but we always could have been done a little bit more, better log thinking earlier. But thankfully everyone's healthy, and thanks, everyone's healthy.
So you also, your company has a pretty famous founder, Jennifer Doudna. Can you talk to me a little bit maybe about how she was supportive and discuss what was happening with the Mammoth team?
Yeah, so she's been very much engaged, and we'll talk a little bit about our work with UCSF, and Jennifer had a lot to help with the background of that, but with Dr. Charles Chu at UCSF and his lab, IGI in Berkeley. But yeah, so Jennifer tends to work in the background with us. Our two co-founders are out of her lab and are amazing scientists. And so there's this whole stream that she does. I don't know where she finds the time. If you know Jennifer Doudna, she's everywhere all the time with a very insightful conversation. So she's there. It's just in the background. I really think that we have to really recognize the experts in infectious disease like Dr. Charles Chu at UCSF and others around the country in Cambridge. There's quite a few experiments going where early outbreaks gave the ability to access patient samples, and that's what this is really about. You can do contrived samples all you want, but it's really about taking the patient sample and testing it.
Cool. Yeah. So looking at this the slide we've got up now, this was another set of perspectives that came out of this golden interim period, really to talk about the fact that a lot of scientists were in this survey that was done by the Linus Group, essentially saying that, hey, I can actually do some work now that I'd put off because I had a lot of other things to do. I can go back, I can catch up on publications. I feel like we're all, especially if we're in the CRISPR space, we're all perpetually catching up with what's the latest and greatest thing. Maybe just read preprint servers or Twitter to get your CRISPR news. And so I guess, you mentioned earlier a little bit about the social impact. How critical is communication for your team when you're trying to progress experiments forward versus doing it face to face?
Yeah, I think it's gone pretty well. I think the publication component, we just published a paper. It was a collaborative. I think those things are in place, and so people were able to work effectively together. But I do think it's still, again, the missing the personal interaction. Maybe you're missing the hardcore science is getting done because it's pretty easy for these people to do it because they've done it in the past. I think it's this lack of social interaction that's actually kind of underlying stress that's putting on. So when people are writing and reading at home and doing that work, they're kind of like, am I contributing enough? Am I doing enough to help the company or help my peers?
And is this, do you feel like the activities that you're doing at Mammoth and your scientists are working on, Janice and Lucas and everybody, do you think it's pretty similar to what you would expect the academics are going through right now, or is it pretty different?
I don't know. I wish I could say in the academic world, because it operates under a little different model. I would think so. I would think that, obviously they're going to put social distancing in place and safety at the universities and academic labs. Grant writing is probably pretty, I bet there's a lot of shifting of grant writing towards COVID-based or, infectious disease-based experimentation, that they're taking their core research themes and theses and moving them towards that to get funding. There's a lot of funding out there, as we know. And so I think that in the academic world, that's probably what's happening, is people are thinking about, how do I pivot a little bit of my research into that? So funding drives everything in academics. And then trying to get some relevant, if you do have some relevant experiments in the COVID space, trying to think about how to write and get it out. We certainly have been challenged with that. There's certainly a hunger out there in the press and for publications around COVID, relevant COVID stories and research and advances.
Well, yeah, speaking of relevant COVID resources, I think now actually might be a good time to actually go into discussing what you've been involved with a little bit. So can you tell us how Mammoth got involved and how you got involved with pivoting Mammoth's detector platform to generate a rapid protocol for testing the SARS-CoV-2?
Yeah, be happy to. Well, let me take a step back about Mammoth and just foundationally, and we can go to the next slide, it's fine. So Mammoth is a protein discovery company that's focused on discovering new Cases. And so we're best known for our diagnostics. That's where we started the company, but really the foundation is around identifying Cases from nature and their functionality. This is really hard. I had no appreciation. I grew up in the world of most people Cas9. And by the time I got into it and met Kevin in 2015, 16, the guide RNA sequences were known. Cas9 functionality was kind of known. The mechanism wasn't completely known like now. There were some interference and activation, molecules, modifications and motifs of Cas9. And it's been such a great first mover into the industry. It's such an MVP. I mean, I don't think Cas9 gets enough recognition for all the things it does. But there are limitations, and the experimental designs and the creativity of the scientists are asking to do more. And so there's actually, because of the power of the Cas enzymes and the simplicity of doing a CRISPR experiment, once you have the basic knowledge, really opens the door up for new uses of Cases. And they go into three groups, and I'll just say the first group is gene editing, Cas9, so they're editors. There are detectors, which are diagnostic based, and so there's that group. And then there's this other group, which are modulators, which are all coming out now and interesting, and we'll hear more about that over the next couple of years, and probably going to be the foundational truth going forward in the 2020s for therapeutics and other applications, ag applications. A little bit about the company, just needed to talk to you about that. So we are doing gene editing, discovering Cases towards that direction. Looking at other enzymes than just 12 and 13 for reporting and diagnostics, and then even looking for other functionality. So diving in, so that's kind of the foundation of the company. if we look at diagnostics, and this is a cartoon of kind of the mode of action, you need a guide RNA to target the gene of interest or the target sequence of interest. You need a Cas protein. In this case, it's libcas12a that we're using, a very common general one. And then you need the third thing, which is a cleavage reporter, a piece of single-strand DNA that has some fluorophore or some reporter molecule in the gold star, and then some other molecule like a quencher that keeps it silent while they're close in proximity. And so, and then lights up once it's cleaved. And so in the middle diagram, you can see you have the Cas, and thank you, Kevin, the RNP, what we call the complex between the guide RNA and the Cas protein, and then you also include the reporter molecule. When it interacts with the right diagram, interacts with the gene of interest with the blue rectangles there, there's a conformational shift in the Cas, and it indiscriminately cuts single-stranded DNA as a mode of interaction, and that cleaves the reporter molecule and gives off a signal. So that's how Cas does something in this diagnostic reporter instead of just editing for knock in, knock out, and all the experiments doing that way. Okay, well, everyone over breath on that one. And
that's a lot to digest.
It's a chunk, but it really is pretty simple mode, because now I'm going to take it a little farther. Okay, and if we get the next slide. I'll keep this a little bit short because I think some people know, but yeah. So we just published last night. It came out in Nature Biotech, collaboration with the UCSF and Dr. Charles Chu. This publication just came out, dropped at midnight last night, around testing of 83 patient samples, COVID patient samples using our what we call CRISPR COVID detector. Which is our version of using our chemistries around the Cas12 and the COVID virus and RNA. And so the workflow is pretty simple and kind of somewhat common to the CDC assay with a couple twists. You take the swab with the patient sample, you extract the viral RNA, and then you do real-time isothermal amplification. We call it's LAMP is the one we're using. And we're looking at the E gene, the N gene, and then a control RNase P. And then you run, obviously, the amplification to get enough DNA in there. It's RT, so converting the RNA to DNA. And then the second step is adding the guide RNA Cas12 complex with the reporter molecule, the single-strand DNA probe, and have that cut. And then you can either measure it one of two ways. You can measure it if you have equipment. You can run it with a fluorimeter and measure whether you have signal or not, or you can run it on lateral flow, which we did in both cases in the study, but really lateral flow is easier. And if you look at the amount of equipment you need to run this assay, is a couple thermal blocks, one for LAMP, one for detector, pipettes, and a lateral flow strip. So there's no PCR equipment in here or that. Now, in a real clinical lab, that's not a problem. But what I'd like to make the point of this, and probably the last point, so I'll let you read the paper and see the results. We've really good sensitivity, good correlation, 95% positive correlation to CDC assay and 100% on the negative. So really good results. We really did this fast. A question just came up that I'll jump on. What's the cost? Right now, it's incomparable to the CDC costs. But when you think about where we're going with this, we want decentralized diagnostics. So one of the challenges, if you think about, and I've worked with the DT a long time, what's the problem you're trying to solve, right? And the problem here isn't necessarily detecting the virus, because that's there. The real challenge is that we're doing it in a centralized lab. So we have to go to some testing facility, drive through, doctor's office, hospital, especially with this nefarious virus, which can be 5 days asymptomatic but infectious, and expose a lot of people to this virus, that if we were decentralized and we could actually test at home with a handheld device and get some feedback that says, Bing, you have the virus. We think you might need to, talk to a telemed doctor to go get help, or it's negative and it's like, okay, you don't have it. that would be much more powerful in the airports, in places like Amazon setting up their own testing labs because they're so frustrated with the decentralized version. And so we think as a company, given the simplicity of the equipment down below, that we can miniaturize this into something the size of like a pregnancy test at home and low cost. We're not there yet, okay? As evolution and new things happen and as startups go, we are going to be pushing hard towards this direction because we think that that is solving a social problem in addition to a science problem, and that's very powerful. But this is a benchtop assay, so we're not so, and it was very robustly done, but it was very quickly done, and I think that there's so many other improvements we can do on it.
Ted, can you actually, so you mentioned it was something that you guys were basically able to, rapidly adapt your tech to do this. Can you talk about how, like, where that decision came from, like, who got involved with that decision, and how quickly you were able to just say, hey, let's go make this, now you have a publication that dropped in Nature this morning. So congratulations on that. But to go from that genesis, that thought, to where you are now.
Yeah, super interesting evolution. So I think there's one key moment that happened that actually flipped the switch. So up through January, we got early access to the sequence like everyone did from Wuhan, and then the Washington virus. And obviously everyone was sequencing all the different strains that were mutated strains that were coming out. And we were like, okay, we can quickly knock out a white paper here. And we posted that very quickly in what was it, early February, on how to actually do this experiment. But it was I'd say a very quick and dirty way without patient samples. It was contrived samples, and with the eye that we would work with Charles to do this. So we were this was the endpoint we wanted to kind of get to, I'd say back in February and even March. Early March was that we wanted to get a paper out that could demonstrate the chemistry is solid and valid. CRISPR can do this, because this was the first time with patient samples really demonstrating CRISPR with COVID. And so something happened, and the big thing was the EUA, the emergency use authorization by the FDA, lowered the bar for requirements, because we're a small company. For us to release an assay and get the FDA to look at it is a big deal. This is a 12 to 18 month process in the diagnostic space. Well, they lowered the floor and said, look, we're so desperate for people to come bring technology to us that can change how we do things, along with the big equipment that comes through the Abbotts and Roche's systems that you see. They're going to actually be the workhorses to solve our testing problems. That's going to continue on, but also lots of companies that had technology like ours that are going out there. Not all of them are that good, but they went through, and the FDA is like, we're going to lower the bar. And I think there's a lesson here for science, and the question for the FDA, which really focuses on efficacy and safety, okay, is how far do they go safe and not? And I think that's a whole different discussion, but and we'll see more as we go on. But when they lowered that bar, it was like, okay, now we can actually work with UCSF and go get an EUA, which Charles talked about in his press release today. And they will support and submit this chemistry with their lab only, obviously, for an EUA. So that was really exciting for us, that all of a sudden now we can talk to the FDA about our chemistry. And now not only is it this really cool piece of equipment down there that that now we can miniaturize, but we actually have some conversations with the FDA, which is really interesting. So the evolution kind of came from the scientific need and the human need, all of a sudden, to an adjustment by a government agency, which was awesome. And then it was like the door opened up when we could actually go ahead. Otherwise, we'd probably be in a 2-year process to get this out. So it really did accelerate our program.
And I guess you must have had to also get some partners aligned really rapidly as well to be able to supply you with the reagents that you needed to do this. I imagine you must have had a great guide RNA company help you with that too.
Yeah, well, Synthego's been amazing. I will say that.
All right.
And I will say other suppliers. And the reality is that everyone understands the need. The beautiful part about this whole negative thing is that it's hit everybody. There's nobody indiscriminately group that's just isolated. It's everybody. So when you have a conversation with a supplier or a service provider or even a friend, they all get it. And so there's prioritization. So I'd say the suppliers have motivated themselves to support the COVID industry, and they've done a great job. I've been totally psyched.
I think Aditya has some questions from the audience. Maybe briefly before we go into those. Our topic today is to keep calm and CRISPR on, to carry on. Any words of advice for maybe either academics or small companies, or even large companies? I know you have some experience with large companies. How to just get in that mindset.
So I think I have 2 college student aged adults at home, young adults at home. One a sophomore, one a master's student that are working or taking classes. And they're feeling a lot of confusion. There's a couple things going on right now. And I'll just say, one is a frustration of them of just the social aspect got ripped away from them very quickly by the social distancing. And so there's a lot of reticence and anger around, why can't I go see my friends? Okay, something as simple as that. But that transcends itself into working from home and trying to figure out what to do. And so with my son, we're very much in the moment of he's getting his master's in mechanical engineering in June. What do I do now? I kind of had a really nice job market in the summer 2 months ago, looked great, now not so great. And so here's a couple of things I'd say. If you're going to go out and look for a job, or you're a small company, think about this. Think about volunteering, okay? Because I don't think it's going to be hard for companies to spend time to hire people that are not well-funded. They're kind of making the adjustments. They tend to move away from hiring new people because the onboarding, the training, all that. And so I would say this, and it's hard. It's a hard answer right now, and it might change in a month. So I'm going to put a disclaimer on it, but think about volunteering, think about interning, or thinking about contributing to the help, like IGI at Berkeley. UC Berkeley was looking for volunteers to help set up a lab. There's going to be, I think, some opportunities to get acquainted. And what this does, it connects you into the industry, and you'll meet people. And I think my experience, in many decades in industry is making relationships with people is the way to get jobs. And so if you're a PhD and you've been working at the benchtop, but you haven't reached out to peers or other people publishing or people in industry you want to get a job, I do it now. I do it through LinkedIn. I do it through relationships or introductions. And I think that will get the foundation in place so that you can go get a job. Because otherwise, it's so unclear what's going to happen the next 6 months in terms of hiring and being able to be current, and maybe just stay in academics for a little while. I don't know.
Okay. So as Kevin said, we're getting into the Q&A part, and we got a great question from Betty. Is the guide RNA designed to recognize the N or the E genes, or both? How does the ssDNA interact with the Cas12 complex?
Yeah, okay, so we actually run 2 channels in this. We test separately for the E and the N gene, so we have separate guides for each one of those. You can multiplex them up, but you can run them separately. Actually, the FDA right now is dropping only to 1 gene for confirmation to call positive, as long as the control is good. So it will come back up to multiple genes. So we run those separately. And what was the second question?
The second question is, how does the ssDNA interact with the Cas12 complex?
Yeah, there's a few papers out there on that, and I'm probably not the world's expert to answer that question. Kevin actually might be able to do a better job than I can. But I'll just, so maybe we can, there's some papers on our website to do that. I'm going to punt on that one so I don't embarrass myself with misspeaking.
Yeah, I think you could just probably say that when Cas12 gets activated, it has this sort of indiscriminate activity. It'll start to cut single-stranded DNAs that are in the vicinity. So
yeah, there's an epitope on the backside of the Cas12 that actually was a conformational shift after the interaction with the guide, and I know a little more than I'm saying, but the guide and the DNA. It actually, the conformational change allows it to go and cut the single-stranded DNA. So it's a different section than the interaction, the epitope that's interacting with the guide RNA and the DNA. But there's some really good papers out there talking about this mechanism now. So there's a lot of work being done.
There was actually another question that came through the chat. It was a question I had as well. So from Zoe, where she said that, what are the challenges of large scale implementation of this? Is any of that exacerbated or alleviated by the urgency of the scientific community during the pandemic?
Scaling, and I've worked at large companies and small, scaling is a thing. It's not easy. And I think that the steps you need to scale are important, because as you scale, you need to de-risk things. In research, you can take huge leaps of steps. You can go as fast as you want or slow as you want, but it's pretty methodical as you scale up. As we're talking tens and tens of millions of tests, okay, and trying to get some level of manufacturing around that, consistency, reproducibility, you can imagine it's pretty hard. And I think that this is something that the big companies are really good at. The Roches, the Abbotts, the Kaigens. And I think that they already have the systems in place. So for a little company like us, we love partners like that to talk to them more, because they already have those systems there. I don't have to create them. And you see with any small company trying to bring technology forward, a lot of times they get bought, or we're not saying we're doing that, but they partner with large companies because they can stick it in their scale channel and then distribute to whatever regions they have approvals or worldwide.
Cool. So we got a few more questions. Karen from UC Davis, how are labs operating during shelter in place?
Yeah, so I think I've talked pretty good about this one. I think it's mainly they're trying to prioritize safety first, productivity second. Yeah, that's probably the short answer. I think I've kind of probably spent more time on that.
And then the second one, Quan, sorry if I'm butchering this, from UAB, what are some ways to keep working and stay productive?
So I kind of mentioned this before. I think building habits right now is, if you're getting up at 6 a.m. or 7 a.m. and getting into work at 8 and working 8 to 7 or 8 to 6 or 5, I would keep on that schedule and trying to do that, and as much as you can. There's this challenge with people who are at home, who have children at home, young children at home, and I have a few Zoom video meetings where I've had young ones pop into the screen, and we're all really we all understand this is life, it's different, and it's okay if that happens. But creating some space and time to habitually keep your meetings going in time of stress, I think is really good. If it's very chaotic and your schedule around it, and we're in the middle of this chaos around you, the stress level picks up and it's hard to focus and be productive.
Yeah, that's really key. I think that's what we started CRISPR Office Hours, and going back to just being empathetic of the situation, supporting one another, as you said, with family, with kids, as we try to be productive and tackle through this. And we got one last great question, I think. And what role will COVID-19 play in the future of biotech?
Yeah, I have a real specific thought about this, and it goes into 3 tranches. So COVID is stressing the systems mainly around diagnostics and testing and around vaccines. Okay, so vaccinations. And the first one is we just can't scale quick enough. We weren't ready. We were caught short of diagnostics. On the vaccine, it's the bastard child of biotech, right? No one really wants to put a vaccine out because they're pennies per dose. It's hard. It's ubiquitous to everybody, so it's really hard to get that's effective to everybody and you don't have side effects. It's really a tough, high bar because of the number of people that get it. And so I think the vaccine game is still just starting up and warming up here, and we'll see how that plays out. But I think COVID-19 is going to bring that forward, and you hear companies like Moderna and others, they're getting into yesterday, I think J&J and GSA, everybody's starting to come in hard to that, and they're going to dump money into it. So I think actually it's going to change the viewpoint of vaccines more than diagnostics, and diagnostics will continue to move forward on front end with molecular diagnostics on early detection, and then with antibody diagnostics for surveillance. Those are well known, but technology evolution. And then there's this third category in the middle of therapeutics. How do we treat people and minimize people that have been exposed, to limit cytokine storm reactions and the upper respiratory effects, and that's still to be played out too. So I think actually it's one of the things that's going to actually hit the three main areas of diagnostics, therapeutics, and vaccine. So totally a big deal.
Yeah, there's one great question I just got, and I said the last one was it, but this one is pretty good from Laura. The news has so many stories about supply side issues in PPE, and now key medication is related to treatment. Do you see challenges with supply chain for this diagnostic test, and that could limit the utilization?
Yeah, I think so. I don't know if it's too much to go back to the chart with, if you guys wouldn't mind doing that just really quick. There we go. So I think what you're talking about is this transition phase. That's really the challenge. The Golden Interim is kind of making the adjustments, but this from the Linus group, I think, is a little ambitious about productivity in the transition phase. I think actually it's going to be longer and flatter, because as we all spin back up and needing PPE or reagents like pipette tips, okay, these factories were shut down for a while. Now they're probably spinning back up, but just for that example, we're all going to start pulling on the supply chain and for the mundane picks and shovels of the industry. And I think that those are going to be the things that we're going to be looking at, the thermos and the VWRs and that, to really stock up so things like that don't hold us back. So I think the supply chain is going to catch us in areas we're not expecting it. I think the access to enzymes, those companies that I've talked to them are so ready for it. So to make the cases, to make their master mixes, and that, I think they're all ready. So I'm not so worried about that. I think actually it's going to be, and maybe I'm wrong, I'll probably be wrong, but the mundane is actually going to get in the way. And just like we saw, who would have thought masks, right? In this world of, where we were 6 months ago, masks would be the rate limiting factor for, transmitting this virus.
Yeah, that's definitely a good point. So we're at time here. I want to thank everybody for joining us. There have been great questions, lots of interaction. We love that. Thank you, Ted. Thank you, Kevin. As promised, how can you guys get your shirt? Go to synthego.com/claim, and I'll fill out the form. We'll send you the details. As always, this recording will be on YouTube. Really excited about the attendance and the interaction every time. And we want to make sure you guys know next week we'll be having Omar and Jonathan, who are both McGovern Fellows from Sherlock, on next Thursday at 9 a.m.
So I'm going to just jump in here real quick. You weren't ready for this, but Jonathan and Omar are amazing researchers, and Sherlock's doing some of the similar things that we are. And I think if you showed up for today to keep calm and carry on and understand a little bit what CRISPR can do in the diagnostic space and other applications, then show up next week for seeing Jonathan and Omar. They're pretty amazing. We're all working towards the similar goals.
Cool. Yeah, thanks. Thanks, Ted. It's really great to mention that. And Aditya, just to correct you, sorry, go back to the t-shirt. If you want to pick up one of our great t-shirts, actually it's synthego.com/com. So
and claim your shirt.
Yeah, to claim it. So to claim it, go to synthego.com/com. So yeah, looking forward to having the two guys from MIT next week as well. So Ted, Tisch, thank you very much for your time. Appreciate it. I know you're super busy, and I think the effort that you've made with Mammoth and with Janice and Lucas over there to get this assay out and into something that's really relevant and timely is fantastic work. So keep up the great work, and we really appreciate your efforts.
Thank you.
Thank you, Ted. And thanks, Kevin, as always, for hosting and correcting my dyslexia. I appreciate it.
You need more coffee. It's okay.
Thank you.
All right, everybody. Thank you very much. And we'll look forward to seeing you next week on CRISPR Office Hours.